Okay. So let's continue. So to remind you what we finished on just an hour ago. So we were talking about the envelope of bacteria, and we figured that a lot of bacteria have cell wall, and it's, outer to the membrane. Membrane, cell wall, gram-negatives have even outer membrane. But moreover, some cells have so-called glycocalyx, which is a layer of carbohydrates, which is the outermost layer in some cells. It's even farther from the center of the cell than the cell wall or the outer membrane. Okay, it's not in all bacteria. Now, glycocalyx can form two distinct structures. It can form capsule. You can see it on this picture. It's shown in a green color. Okay, and you can see capsule as the white halo around the cells on this electron micrograph. And capsule is a pretty firm structure that helps cells to do two things. First, it helps them to adhere to the surfaces. Okay. Second, it helps them to survive phagocytosis. Now, to give you a clinical relevance of what capsule is. So capsule, is that a trait? Is that a phenotypical trait of a bacteria? To have, yeah, I usually, look, questions that I ask are usually very simple. Simpler than you think. So if that is a phenotypical trait, it's the result of what? Phenotype is the result of expression of genes, genotype, right? So there should be some genes, right, that encode ability to form capsule. Do I sound logical so far? Now, it turns out that if you will take a microbe, for instance, that's a famous Griffith experiment. You take a microbe, Streptococcus pneumonia, bless you, Streptococcus pneumonia, that causes severe disease in humans. You can stick the same microbe in mice, and mice are going to die. This microbe has capsule. Okay? If you manage to remove genes responsible for the capsule formation out of the microbe, what do you think is going to happen to the ability of microbe to kill the mice? You remove capsule, microbe becomes, okay, so is it going to kill mice or not? Probably not, and it turns out it's true. So if you take a strain of Streptococcus pneumonia that does not have a capsule, it's non-pathogenic in mice. Does that make sense? So that is another example of virulence factor. That's another virulence factor, capsule. Okay? Another type of glycocalyx is slime layer. It is looser, more loose than capsule. Okay? It helps bacteria to stick to the surfaces very well. Moreover, it helps one bacteria to sort of stick to other bacteria, forming what we call biofilm. Biofilm is the bacterial community in which different species of microbes live in a mutual or synergistic relationships. Now, what is mutual relationship? What is mutualistic relationship? It's correct, but elaborate on that. What?
Either the bacteria would probably be either like the same type or like attempting to achieve like the same end product. Or
Okay, that's one version. That's actually you stepping farther from that. So when bacteria in mutualistic relationships, they help each other. They not necessarily have, all living organisms have one goal, which is what? Stay alive and reproduce. That's only one goal for all living organisms. So when bacteria are in the mutualistic relationships, they somehow help each other to do that. Now, mutualistic relationships are what we call, they usually obligatory. Two mutualistic bacteria cannot survive without each other. Does that make sense? They just can't. Now, synergistic relationships. What do you think it is? What is synergism? Elaborate. Just hand in hand? If you drink a shot of vodka, you're going to be buzzed. If you drink a glass of beer, you're going to be buzzed. If you take a shot of vodka, pour it into the glass of beer, and then do this, and then drink it, what's going to happen to you? Huh? Oh, it's going to get you, it's going to knock you off your feet. Don't do that. Okay? It works fine. I mean, you can get wasted from much smaller amount of alcohol, but that's synergism. Okay? When bacteria are in the synergistic relationships, they can survive individually. But when they together, each of them is more successful. Does that make sense? They don't have to be in that relationship, but they are more successful when they have it. So microbes live in the communities mostly. You have biofilms on your skin. One of the biofilm that you can actually feel, you can touch, go to the river. What is it, a Chagrin River, right? That flows next to the campus. And walk barefooted on the rocks in the water. How do they feel? Slimy? That's bacteria, fungi, algae, all kinds of microorganisms growing as the biofilm. That's really a biofilm. It's a great example of it. Okay? So both capsule and slime layer contribute to the biofilm formation. And capsule additionally protects against phagocytosis. Why biofilms are so great? First, nutritional chains. One microbe can produce nutrients that are utilized by another microbe. Okay? Does that make sense? Microbes, when they form biofilm and they form that common slime layer, that slime, they protect each other from drying, which is the scariest thing for bacteria, to dry out. Okay? Why it is so clinically relevant? Because of this. Biofilms in a hospital grow on virtually everything. They can grow on catheters. They can grow on stethoscopes. They can grow on prosthetics. Imagine prosthetic joint. It's really hard to keep it 100% sterile. Seriously, just 100%. I mean, not a single cell. So when it gets implanted, let's say prosthetic hip, there's a good chance that there are some microbes in the joint. And if they start to grow, they form biofilm on that joint that will cause inflammation of the joint, right? Well, the first thought to do is to treat it with antibiotics. The problem with biofilms is that they are quite impermeable for antibiotics. Bacteria in the biofilm survive antibiotic treatment really, really well. It's a huge problem. Okay. So often people who receive prosthetic joints have to be on the prophylactic antibiotic regimen just to limit the bacterial growth. You know, because while you keep it under control, it's fine. When they establish biofilm, it's sometimes people have to have the joint replaced just because of the bacterial contamination of it. There are some new treatments with a very small electric current that seems like it's much better in killing the bacteria. The only issue now is how to deliver it to the side of the joint, because you don't want really to electrocute the person, who just received a prosthesis. In archaea and some bacteria, GLYCOCALYX is represented as the S layer. We don't really know what they are. It's some other sort of glycocalyx in which you have both carbohydrates and proteins. Maybe they make cell a little stronger. Okay, so take home message: understand what glycocalyx is made of, carbohydrates, and two basic types, capsule and slime layer. Why? What is the function of each? Clear? External appendages: flagella, fimbriae, and pili. Flagellum. That's a heck of a picture, isn't it? No, you don't have to know the structure of the flagellar motor. Okay? You don't have to, really. Basically, you have, in both gram-positive and gram-negative microbes, you have a rod, okay, that rotates, and the flagellum, the filament of flagellum itself, is attached to the rod via the hook. So what happens is something like that. Okay, see? Flagellum rotates and microbe flows. Obviously, rotation of that sort requires quite a lot of ATP, right? And ATP actually feeds, in some cases there is, as far as I remember, there's hydrogen flow involved into the flagellar movement. Okay. So yeah, they create proton gradient. Okay, and then protons flow through the membrane, and essentially it's this. You have a proton diffusion. Okay, they diffuse, pretty much rolling down the slide, and as they diffuse, they release energy, and that energy feeds into the flagellar movement. Does that make sense? Different bacteria have different arrangements of flagella. Monotrichous is when you have a cell with a single flagella on one pole of the cell. Amphitrichous, flagella on opposite poles. Lophotrichous, bunch of flagella at one pole. And peritrichous, flagella all over the cellular surface. Before we move on to the fimbriae and pili, what you have to know about flagella, the idea that it requires a lot of ATP that generates proton gradient. Okay, and proton gradient immediately provides energy for the movement. And 4 types of flagellar arrangement. Does that make sense? Flagella, is that a phenotypical trait? Yes. So it is encoded by certain genes. Flagellum, which advantage does it provide to bacteria? Ability to move. If bacteria can move compared to its counterpart that cannot, moving motile bacteria, do you think it's going to have higher or lower virulence? Higher, exactly. Flagellum is another example of a virulence factor. Does that make sense? Okay, let's move on to the fimbriae. I don't have a picture for it, but it's a very, very short, like velvety structure, like a brush of protein projections on the surface of the cell. Does that make sense? Think about it as the Velcro for bacteria. Fimbriae helps bacteria to attach to the surfaces. You're gonna get tired of that statement. Is fimbriae a phenotypical trait? It is. So it is encoded by what? Genes. When bacteria has fimbriae and can attach to surface, compared to counterpart that does not have fimbriae, which one is more virulent? With the fimbriae. Does that make sense? It's another virulence factor. All right? Now, pili. You can see them on that electron micrograph. Two bacteria apparently connected by pili. What do they do through the pili? It's called conjugation. Yes. Microbiologists usually say that bacteria have sex. In a common sense, they don't, of course, because bacteria are asexual organisms. They do not have male or female. But what is the main purpose of sex in animals? Why animals do not just clone themselves? Like, you know, you have a deer and then a little deer buds off the side of it. Why? Yes, genetic diversity, okay? In mammals and fish and in animals, sexual reproduction establishes genetic diversity. Bacteria, how do they reproduce? We didn't talk about it, but what we have one bacterial cell. What happens to it when it reproduces? Splits into, and those two bacterial cells, are they similar to the original one, to the mother's? Well, they're actually identical. There's not a lot of diversity, genetic diversity going on here. Why do we care so much about genetic diversity? It's the mechanism of fundamental biological process. Yes, exactly. It's responsible for evolution. So, if there is, I mean, mutations are fine, but even mutations accumulate rather slow and gradually. So without the exchange of genetic material, bacteria wouldn't be able to adapt effectively to the changing environment. Does that make sense? Pili provides such mechanism. Pili can connect to bacteria, and bacteria can exchange parts of the chromosome, or they can exchange extrachromosomal DNA. Does that make sense? Right? By the way, did we talk about extrachromosomal DNA? We did not. Let me go back for a second. We should have, but we didn't. Oh yeah, we did not. We're going to talk about it later. Not now. Okay, so pili allow bacteria to exchange genes, and if it's a gene that confers a trait, say a trait for antibiotic resistance, this antibiotic resistance will spread through the entire population. And that's actually what happens all the time. One of my favorite examples of this is the study that was done in Netherlands. They had several cases of antibiotic-resistant E. coli infection in humans. And they were trying to trace down these microbes. Where did they come from? Well, guess what? They came from poultry. They came from chicken. Poultry used to be regularly, prophylactically treated with antibiotics. It improved the growth rate. Well, bacteria adapted to that, and then at some point jumped into the humans. But the point is, there was one strain of E. coli that had that resistance gene. Turns out that this strain of E. coli managed to transfer that gene to apparently different species, Klebsiella pneumoniae. So that antibiotic resistance goes through the population and between populations of bacteria, and that's actually a huge threat to the healthcare now. the spread of antibiotic resistance. You always have to think about it. I mean, not always, not every minute of your life. Now, how cells move by flagellum. They do two types of movement. It's called, one movement is called running. Okay? When cell moves, swims straight, and then it stops and starts to tumble. Okay, right, and then it runs again, and then it tumbles again. Does that make sense? It's called run and tumble movement. Now, bacteria, do they have directionality in the movement? Remember that the plate with bacillus that we observed, in a couple of plates had bacillus colonies. And those colonies were pretty big. They were growing in diameter. Why? Bacteria were moving from the center to the periphery. Why? What were they moving towards? What was outside that is not inside anymore? Nutrients. They ate everything inside and they were moving outside. Does that make sense? They were going towards the higher concentration of nutrients. This movement is called chemotaxis. Does that make sense? Towards the increased concentration of nutrients. Does that make sense? To the increased, say, you're a bacteria, you swim where the nutrients are high, it's called positive chemotaxis. You have to understand, bacteria don't just swim like Michael Phelps in one direction. They go actually quite chaotic, but the general idea is to move towards the higher nutrient concentration. Does that make sense? If bacteria tries to avoid some kind of chemical and to swim away from, I don't know, a toxin, it's going to be negative chemotaxis. Does that make sense? Swimming away. Now, can bacteria swim towards the light? What do you think? Sure, why not? Especially if they can photosynthesize. In this case, they need the light. So how would we call movement swimming towards the light? Phototaxis. Remember we discussed the bacteria that kind of orient themselves along the magnetic lines of magnetic field. How we call this? Movement along the lines of magnetic field. It's magnetotaxis. It's magnetotaxis. So taxis is movement in this case. Does that make sense? Got it? So you have to understand run and tumble concept of movement. It's not like straightforward. It's only a general direction. And second, concept of chemotaxis, phototaxis, and magnetotaxis. Just recognize what they are. Recognize what these words mean. Okay? We good? Eukaryotic cell. There's a shitload of organelles here. And I actually will leave these squares for you to fill up. It's going to be like your exercise. It'll help you because we're going to discuss all those parts of the cell separately. What I want you to notice is that compared to the bacterial cell, it's more complex and it has much more membranes. In fact, it's more wait a minute, I'm going to say this word. Compartmentalized. which means it's separated in parts. Does that make sense? Let's see which parts. Well, first, most obvious, it's the nucleus. Okay, nucleus is not considered a cellular organelle. Nucleus is a nucleus, it's not an organelle. That's where the DNA is stored. You can see this on this picture. It looks like a bunch of spaghetti. Okay. Nucleus is interesting because it is surrounded by the double membrane. Not just one membrane, but two layers of membrane. Does that make sense? You have two layers. These two layers of membrane form nuclear envelope. Nuclear envelope essentially it performs a structural function. Okay, it shows you the boundaries of a nucleus. Nuclear envelope has so-called nuclear pores. Unfortunately, in this picture, it's not. No, it's in. That's a nuclear envelope here, and that's a nuclear pore. What is the function of nuclear pore? What do you think? So allows the genetic material to pass through. Which genetic material? Say again? RNA. DNA doesn't leave. Remember, think about that freaking cookbook. Your cookbook never leaves the precious bookcase in your living room. It's a family relic. It should stay there. You don't take it to the kitchen where you can spoil it or burn it. Okay? But RNA can go back and forth. In fact, if DNA is not in the nucleus, but in the cytoplasm, what does that mean? Something's fucked up in the cell. It really shouldn't be there. It must not. If DNA is in the nucleus, is in the cytoplasm, something's wrong. Okay? So you're right. Pores allow the passage of the material, the RNA, because RNA is made in the nucleus. And then it goes into the endoplasmic reticulum, which we're going to talk about. Inside of the, now, nucleus is of course membrane bound. Inside of the nucleus, you can see that dense assembly of proteins mostly that form nucleolus. Nucleolus is responsible for synthesis of ribosomal RNA and assembly of the ribosomes. Nucleolus, as the part of nucleus, doesn't have a membrane that surrounds it. And here we go. Nucleolus, when it produces ribosomal RNA, that ribosomal RNA will leave nucleus via the pore. Okay? Now, when DNA is used as the matrix for the synthesis of RNA, how do we call this first step, by the way? Transcription, yes. So after transcription, where does mRNA go? Where does it go? What's the next step for mRNA? What does it do next? It's getting translated into what? Protein. So where does this process happen? Where does the translation happen? In the endoplasmic reticulum, okay? Which part of it? rough. So rough endoplasmic reticulum is where the protein synthesis happen. Sorry. Why it is rough? What makes it rough? Ribosomes. There you go. So endoplasmic reticulum is essentially a network of the membranes. Okay, it's like a maze of membranes. Part of that maze has ribosomes. That's where the protein synthesis happens. Other part of the same maze does not have ribosomes and is called smooth endoplasmic reticulum. Smooth endoplasmic reticulum is where the lipid synthesis happen, where the carbohydrate synthesis and carbohydrate metabolism happen, and where detoxification happen. Now, if we want to step away a little bit from microbes and look at the bigger animal, like human, which organ is responsible for actually destroying toxins? Liver. Not filtering, destroying toxins. It's liver. How can you kill your liver? Huh? Alcohol. It's a toxin. Very powerful poison. If you would take a sample of the liver of the person who suffers from alcoholism, liver that already has pathological changes, and make a slice and put it under the microscope, this one organelle will occupy a lot of space. It's going to be smooth endoplasmic reticulum. It has to increase in size in attempt to detoxify all the ethanol that goes into the liver. Make sense? Okay, let's move on. Golgi. I'm going to just call it Golgi. Golgi was an Italian. It was a dude. Okay. So Golgi apparatus modifies proteins, modifies lipids. Protein modification has so many versions. Proteins can be glycosylated. What does glycosylation mean? What is being added to the proteins? Sugars. So you can add sugars to the proteins. Lipids can be added. There's no actually specific word. So you can form lipoproteins. Okay. Now we call glycoproteins, it's where it's a big protein and a little bit of sugar. If it's the reverse, big sugar with a little bit of amino acids, it's called proteoglycan. Okay. You can combine sugar and lipid and form glycolipids. If protein is not functional, if it's bad, if it's defective, Golgi apparatus will label it for the destruction, so-called ubiquitination of the protein. There's so many ways to modify the proteins. Okay, so Golgi is unique for eukaryotes. Eukaryotes are very good at protein modification. Now, think about this. Rough endoplasmic reticulum makes proteins, but they're not quite ready yet. These proteins are packaged into the vesicles and go into the Golgi, okay, where they fuse with the Golgi apparatus and proteins being modified. Now you have proteins ready to work. Where do they go? Proteins from Golgi complex, where do they go? I told you, ask simple questions. Where do these proteins go from Golgi? For instance, they can go outside the cell if that's a secreted protein. Can they go to the membrane and work at the membrane? If it's the function of the protein, yes. They go wherever they need it, okay? They go to their destination, okay? But Golgi is rather small, and the cell is rather big. How do they get, let's say, how does the protein get from the Golgi to the membrane, to the opposite pole of the cell? in vesicle, in a transport vesicle. Okay? It's pretty much, it's a membrane-bound bubble that buds off the Golgi complex and then is moved across the cell. Does that make sense? So that's your secretory vesicle. Now, two other vesicles, bubbles, that can be found in eukaryotic cell are lysosomes and peroxisome. Lysosome does lysis. Right? Make sense? It breaks down things. If it's a food particle, it can break down food. If it's a part of dysfunctional organelle, it can break it down. If it's, I don't know, a pathogen, it can break down a pathogen. Does that make sense? How would you break down things in the cell? What would you use chemically? Acid. You'd use acid. I was about to say, think about mafia movies, how they get rid of the dead bodies. Okay, acid. Actually, there's a hydrochloric acid in a lysosome. What else would you use? In your digestive tract, how you break down the food? Enzymes, yes. Proteolytic enzymes, proteases, nucleases, lipases. Okay. And there's another thing, so-called reactive oxygen species. Most of you, maybe everybody, has a reactive oxygen species standing in the cabinet where you store all the drugs and, you know, all of that. What is that? Huh? No. Well, when you store all the medicine, there's a reactive oxygen species. Use it to treat your wounds. Hydrogen peroxide. Okay. There's a ton of hydrogen peroxide in lysosomes. They use all of these three components to destroy whatever they get in touch with, to break it in parts. Okay. Peroxisomes also have hydrogen peroxide, and at the same time they have an enzyme called catalase, which neutralizes hydrogen peroxide, so it doesn't kill the cell. Okay. The function of peroxisome is somewhat similar to the function of the lysosome. They break down things, and they also participate in the biosynthesis of fats and sugars. Okay, you can see the little tiny peroxisome here. Okay, I mean, they're not really fascinating in terms of the structure. It's a bubble, membrane-bound bubble. Now the membrane is not very much different, or not at all different, from the membrane of the prokaryotes, except that its fluidity, its how rigid it is regulated by sterols, not hopanoids. Okay. So membrane is phospholipid bilayer, contains sphingolipids. Let's assume these are sphingolipids. So sphingolipids are the special type of lipids that presumably participate in the signaling in the cell. And it also contains transmembrane proteins. Okay, that's not transmembrane. Okay. Transmembrane proteins, they penetrate through the entire membrane, and they can be a part of the transport system like this one. Okay, they can be channel or carrier. Okay, so that's going to be transport. Or they can serve as the signaling molecule. Now, a few words about the cell signaling. For this specific protein, the one that we just circled, this one, what would be the trigger for initiating the signal inside of the cell? What can be the trigger? What can start the signal? Okay. Chemicals. What do they have to do? It's just what do they're just dancing next to the protein or what? Bind. So in this case, protein serves as the receptor, and the chemical serves as the ligand. It's a ligand receptor interaction. Does that make sense? You've in AP2 you saw a ton of it, especially in the hormone signaling. Okay, you have insulin, it interacts with an insulin receptor. Epinephrine, it interacts with epinephrine receptor. Same goes for practically all of them. For some of them, actually, for some of the transmembrane proteins, chemicals can be much simpler. They can respond to changing acidity. Okay? Does that make sense? But the point is, transmembrane proteins sense changes in the environment inside of the cell, and then signal those changes to the inside of the cell to produce some sort of a response. Got it? Okay? That makes sense? Now, cytoskeleton. What is its function? Structure. Yes. Protein made filaments. There are three major types of the filaments in the eukaryotic cells. First is microtubules. Microtubules are dynamic. What do I mean by dynamic? What do you think I mean? They change. So they get disassembled and reassembled again. They constantly recycle. Does that make sense? The function of them is they facilitate cell division. The mitotic spindle and centrioles pretty much made of microtubules. Okay, they play important motor function, the transport inside of the cell, changing the cell shape, like in amoebae, to enable them to move. Now, we just mentioned that microtubules are dynamic. They get disassembled and assembled again. What if you somehow can stop synthesis of microtubules? What's going to happen to the cell? Yeah, it's going to die. It's going to crumble and die. Well, why do you think I asked you this question? There is a drug that does it. So if it inhibits microtubules, against which organisms this drug will be used? Prokaryotic or eukaryotic? Eukaryotic. The name of this drug is, there are actually two related ones, mebendazole and thiabendazole. And those drugs are used to treat helminthic infections. Especially infections by tapeworms, segmented tapeworms, like beef tapeworm or pork tapeworm. Have you heard the name of the disease cysticercosis? Some of you haven't. It's not really common in the United States. It's a disease in which larvae of the tapeworm crawl out of the intestines of the human and crawl into the surrounding tissues. Okay, if they crawl into the muscles, it's a myocysticercosis. It's a muscle pain, fever, not really pleasant. But if these guys get all the way into the brain, it's said in developing countries, actually, neurocysticercosis is the leading cause of epilepsy in people, because the pork handling standards are far from perfect. a lot of people are infected. We will discuss it, you know, but it's just to give a thought. And then when people are treated, and treatment is fairly simple, it's not a long course of diabendazole, and those larvae and tapeworms, they die. So it's very effective. Okay, no microtubules, they die. Microfilaments. Okay, you can. So that's the picture actually of the microtubules. Use red. Okay, it's microtubules. You can see the green staining of the microtubules here. Okay, does that make sense? Now, microfilaments are made of the protein called actin. You can see actin here, microfilaments, red. So by the way, that's those two pictures, both of them, is the example of the technique called immunofluorescence that we talked about already. Does that make sense? So microfilaments are also dynamic. They assemble and disassemble, assemble and disassemble. They also are involved in the cell division and the movement of the cells, the motor function. And the third type of the filaments is the intermediate filaments, made of the protein called keratin. Now, the place where you can find keratin is your hair, actually. Okay, it's hair and skin. Intermediate filaments are stable. They don't really disassemble and reassemble back again. So the main function is to maintain the cellular structure. Not to change it, but to maintain it intact. Does that make sense? As usual, the take-home message: functions. What are the functions of transmembrane proteins? What are the functions of sphingolipids? What are the functions of plasma membrane? Transport, mostly. Filaments, what are they made of? I may ask you, what are they made of? Microtubules made of tubulin. Intermediate filaments made of keratin. Microfilaments made of actin. Okay, and what do they do? Am I clear in expectations? Good. Mitochondria and chloroplasts. Mitochondria is endosymbiont. Remember we talked about it. What is that originally? What was that originally? A bacteria. Yeah, it was a bacteria that was able to carry out oxidative respiration. It still carries out oxidative respiration. By the way, I saw somewhere the card. Well, it has DNA, it's bacteria. Turns out that mitochondrial DNA is passed from generation to generation, in case of sexual reproduction, only from the mom. Okay. And I saw somewhere the card with the phrase something like, "Mom, thank you for the only thing that, you know, the something that only you can give me." A picture of mitochondria with the DNA. That's actually, and for the higher mammals, for humans, all humans were traced to one woman. So we have primordial Eve. We're all descendants of one female. All of us, all humans in the world. We have mitochondrial DNA of one woman. Anyway, carries out aerobic cellular respiration, which produces ATP, right? Since cellular respiration is aerobic, what does it require? Oxygen, there you go. Okay. This, well, the mitochondria has two membranes. The inner membrane that forms these folds called cristae. Okay. This inner membrane is actually that old, billion years old bacterial membrane. And the outer membrane came from the eukaryotic cell itself. So it has two membranes. Which other part of the cell has two membranes? Mitochondria has two membranes and part. I didn't say organelle. Part. Nucleus, yes. The nucleus has two membranes, right? Now, chloroplast also has two membranes. And inside of chloroplast, you can find stacks of so-called thylakoids. Stacks of the thylakoids called grana. Granum is the singular form. Grana is the plural. So these thylakoids contain photopigments and photosystems that are necessary to carry out light-dependent what? Photosynthesis. Okay? Both of them are endosymbionts. They both can replicate outside of the cell, host cell. You can put them in the egg, as I mentioned. They both have DNA. They both have ribosomes. Okay? Now I have a little question. So is that clear? The function, ATP synthesis for mitochondria, photosynthesis for chloroplast. Good. Can you imagine a eukaryotic organism, single-celled, I will tell you, single-cell eukaryotic organism that does not have mitochondria? Well, imagine that. Now tell me, in which environment such organism would live without mitochondria? It's not photosynthetic, no. Just giving you a heads up, no. In which environment such organism live without mitochondria? Think about the function of mitochondria. What does it do? It's written right there. Mitochondria does what? Is the result of what? Be more exact at the name of respiration. Aerobic. There is no mitochondria. Huh? Absolutely. There is an organism that you may have heard about called Trichomonas vaginalis. Okay? It's the protozoan that causes vaginal infections called vaginitis. Pretty common infection in the United States, actually. Trichomonas survives in anaerobic environment in the vaginal mucosa. It does not have mitochondria, but it has different organelle, which is very exciting. It's called hydrogenosome. It produces hydrogen. Why it is so exciting? What can be so exciting about a sexually transmitted disease? Why it's so exciting that it produces hydrogen? How can we use hydrogen? As a fuel, there we go. So we haven't been able to kind of, how to say, get it to work like in industrial settings, but there's a work, okay, that is aimed to make biologically produced hydrogen. Because if we can make a ton of hydrogen, we can do hydrogen-powered cars and so on and so forth. Does that make sense? It's pretty cool. We don't have to use Trichomonas vaginalis per se. There are other species of the same genus. We can use them. Some microbes have kinetoplast, which is actually responsible for movement. The example of the microbe that has it is Trypanosoma cruzi, causative agent of the Chagas disease. Okay, if you donate blood and you go through the questionnaire before donating blood, one of the questions, I did donate blood. One of the questions is, did you ever have Chagas disease? So they ask you if you had Trypanosoma cruzi infection. Extracellular matrix, flagella, cilia, extracellular matrix. Proteins, carbohydrates. Not on this picture, we don't have it here. You can see the carbohydrates, okay? Proteins and carbohydrates, okay? Forms capsule. There are certain species of fungi that have a capsule. For instance, I think it's Cryptococcus neoformans, agent that causes skin and CNS infections in immunocompromised patients. Cryptococcus neoformans often forms capsule. And as in the bacteria, capsule in eukaryotes helps with the attachment and protection against the phagocytosis. Clear? Okay. Now, flagellum. Flagellum consists of we've already heard that name, microtubules. And flagellum is responsible for the movement. Okay. The wonderful organism that's depicted here, called Giardia lamblia, you don't have to know the name, Giardia, positive agent of beaver's fever. Other name for it is hiker's fever. Has several flagella. Okay. Shorter projections that are called cilia. Usually cilia are all around the cell. Okay, like in this electron micrograph of paramecium. They're shorter, they're more numerous, and they can serve for two purposes. One purpose is to move the cell. Like on the ancient sheeps, a lot of rowers, just moving the cell. Or another purpose is to, by the swiping movement, to direct food into the mouth parts of the cell. Does that make sense? That's the function. We good? Any questions? So pretty much structure, like nucleus, function, nucleolus, function. Okay, you will get this quiz here and figure it out. Let me see if it's a proper quiz today. Yes, it's a proper quiz. It's the right version. Okay, so we're gonna